Okay, guys, so we have started the recording. We'll begin today's session. Today it's going to be Introduction to NCBI. You can go to Google search bar and you can write NCBI, and you will have this in the search results. And click on the first link, which says National Center for Biotechnology Information. Everyone done? Just to confirm if everyone is on the same page. Okay. Yeah, so this is the official website of United States government, USA, America's website, and it has, a lot of databases together. So here in the search, it says all databases, and if you click on this, you have the drop-down menu, and you can see the different databases that it contains. It has structure, it has taxonomy. Uh, can anyone let me know what is taxonomy? I think we learned this in 11th, if I'm not wrong. Anyone, guys? Yeah, correct. If you guys remember, yeah, Rimpai and Bhavya, you guys are correct. So once the organism is identified, you classify the organism. There is this kingdom, phylum, division, class, order, family, genus, species, that thingy. So you have different databases. You have the important ones. You have like gene. It is the database which stores all different kind of genes. And gene is nothing but a fraction of the total DNA which is responsible for a particular function. And you have the whole genome here, and you have GEO datasets, which is nothing but omics dataset, if you guys have heard of. And you also have protein database. You also have SNP. Anyone has heard of SNP? Okay, no worries. So this NCBI, it contains a diverse collection of like nucleotide sequences. It can be either entire DNA sequence, or it can also be a fraction of the DNA sequence. For example, it has Homo sapiens DNA sequence, where it has, the whole genome sequencing of 23 pairs of chromosomes. It also has, for example, E. coli genome, or like mitochondria. Mitochondria has a genome, right? Yeah, correct. Samrat Bhaiya, correct. Single nucleotide polymorphism. Can anyone let me know in the chat what is the cell organelle apart from mitochondria that has its own um genetic data or a DNA? Apart from mitochondria, I think there is one more cell organelle, chloroplast, correct. Yeah, mantra rimpai, you guys are correct. So that is one where it has complete genomes, and the other one is it has genes or gene fragments. There is something called as individual genes, and also there is something called as partial genes. This is homework. Please look into what is a partial gene. So it contains short fragments of genes, or short fragment of the whole genome, which was submitted during the process of research as support of research. For example, if a scientist was doing a research on a particular disease and he used a particular gene from a particular organism, he will submit that during the process, and that gene sequence will be collected here and it will be stored within the gene database. The best example is BRCA gene for breast cancer. I think you guys, it is the famous gene. And then there is transcriptome studies. It also contains data related to transcriptome studies. Anyone heard of what is a transcriptome study or what is a transcriptome? Have you guys heard of uh, reverse transcription in retroviruses? Yeah. Uh, and also, from mRNA, it is possible to convert it into complementary DNA using reverse transcriptase enzymes, right? And this mRNA and cDNA database is nothing but the transcriptome database, where you have the sequence of the gene or the genome where you have only exons. Everyone is aware of intron and exon, right? Intron is the non coding region within the DNA, where exon is the coding region within the DNA. After the transcription, post transcription changes that take place, introns will be cut and exons will be joined, and that process is called as splicing. So, when you have the final polished mRNA, that mRNA and also mRNA converted to cDNA will be stored in the transcriptome studies databases. It also has that. And it also has some synthetic constructs. Have you guys heard of vectors or plasmids? Yeah, I think you will also learn more about this in genetic engineering course. So sometimes scientists they design plasmids suitable for a particular process. Like they take a fragment from this natural plasmid and they take one more fragment from another natural plasmid and they join it together to form a synthetic plasmid. And that also the data of the sequence of the plasmids sequence present in this plasmid that is all stored within the synthetic constructs. And this NCBI data it is widely used in different research. It's used in like genetic engineering research. It's also used in drug testing research and etc etc. It also has some environmental samples as well. For example, if microbiome was collected from soil of a particular region, that is also stored here. And it also has some raw submissions, and it is called as unannotated raw submission uh, because it will just be either like a protein sequence or just be a gene sequence without its classification details or naming. For example, you have the sequence, you know that adenine, thymine, guanine, cytosine, etc etc, a long sequence. You have thousands of base pairs, but it is not mentioned to which organism it belongs to, or what is the role of that gene, or what is the name of that gene, or location where the gene is present. So all these details are not mentioned, and this is called as unannotated raw sequence, where identifying and naming the sequence should be done, but it has not yet done, and that is also stored within the NCBI. Any one of you used PubMed? Have you guys heard of PubMed? Okay, so PubMed, it is like where you get like papers. Like if you search breast cancer and you have different review articles, research papers, as it says free article, You can see you have the title, you have the abstract, and if I want to access this, it says full text link. Let me just click on this and check. Yeah, I've been redirected to a different website, MAG Online Library, and here I have the option of PDF. Are you guys able to follow the steps? You have got the research review paper or research paper. Not sure. You have got this. So PubMed is where you have this collection, or it's a database of research papers or review papers, and you can search for a topic and you can find the papers. Not all papers are freely accessible. Sometimes you might have to have university access, and you also have advanced option. We will look into all these things in detail. I'm just trying to introduce you to different sections of NCBI. You can have filter better by using this advanced search builder. Are you guys able to follow the steps? Okay. Now, any one of you heard of blast analysis? Okay. This is more or less similar to, um, aligning two different genome sequences. You try to find where is the difference between the two genome sequences, where is the similarity. So you find like percentage of similarity. It can also be between protein. So you have two different blast. One is protein blast, one is nucleotide blast. So if I click on nucleotide blast, this was also, I think, one of the assignments. So you have one where you enter the query sequence, the sequence which you want to test, and you also, you know, mention the reference sequence, and then you click on this blast, and this blast will run for some time, and then you will get the result. We will look into all these things. Today I'm just trying to give you the overview of, what NCBI contains. And you have nucleotide, which is nothing but it contains genes and genomes. You can search here. If I want to search something related to, have you guys heard of cystic fibrosis? I think it's also one of the famous conditions. So I'll just write CFTR. It says like Homo sapiens CFTR, which is nothing but I'm trying to find nucleotide sequences in human samples. And the disease of the sample should be deceased and it should contain this cystic fibrosis. You can see I have got different genes. It says human cystic fibrosis gene exon 13. Are you guys able to understand? It says only 47 base pairs are present and it's the exon 13. And you have also exon 4 and it has 49 base pairs. And under every result, you also have different links. One is protein, PubMed, taxonomy, which is not clickable. Oh, it is clickable. And you also have gene bank, FASTA, and graphics. Gene bank is again a database where it's like a storage of genes. And FASTA is a format where you have the base pairs or the sequence of the gene. If I click on FASTA, you can see I have got this as the result. Here, it's a format, and the first line, this is called as annotation. Are you guys able to understand? Here, you have human cystic fibrosis gene, which is the name of the gene, and it also says that the base pairs present in this file are belonging to exon 13, and it has base pairs here. You have CCACA, like cytosine, adenine, guanine, thymine. Are you guys able to understand? And this is called as accession number. And here also you have a bar where you can, filter if you want the same CFTR gene but from a different species sample. You want to look for the same in plants, in fungi, in protists, in bacteria. So you can go for that. And also you have the genes from different source databases, from INSDC gene bank database, from REF sequence database. And you also have different sequence types, and you can set how much should be the length of the search result if you want the search result to contain minimum 1000 base pairs. So you can set all those things here. And if you want to look for the genes that were deposited to the database recently, you can also, set this release data range or revision data, and you can look for that as well. And here it's also, giving you what are the organisms that, have more number of genes deposited here related to CFTR. It says Homo sapiens is the top organism, and then Pseudomonas. Oh my god. Yeah, it says Pseudomonas, then Acromobacter, and then Candida albicans. And you also have synthetic construct again. This is what I mentioned while explaining you the types of different genetic data that this database contains. The good part is most of this data is free, so that even if you want to do a project by yourself and you want some genetic data, you can download it. So that is the good part. And if you click on this graphics, let's click on graphics. Let's look at some simple ones. Yeah, let's look at this exon. So you have two strands. So as you have thymine here, you know that it's not RNA, it's DNA. Am I right? In RNA, you have uracil. So as D is present, it's DNA, and you have two strands, which is nothing but it is representing it as double stranded. And if you click on this, you will get some data. It gives you the location of the gene, and it also gives you the length of the gene. NT is the short form of nucleotide, and with accession number, it is giving you some position. And it has also given you few links if you want to apply blast on it, or if you want to check the genetic record from the gene bank. So you have different. If I want to download the FASTA sequence, because if we want to work in R, we can access it by writing a code where it fetches the gene from the gene bank directly, or you can download a file. You can see that I have downloaded one file. This can be opened in R. Have you guys attended R programming classes? Okay, so yeah, this can be opened in R programming as well. And here you have got some data, and this is also called as metadata. So all these things which are not really used in analysis, usually, like they focus more on the nucleotide part, but this is called as metadata and it becomes very important sometime. It also has, you can see the title of the paper, if I'm not wrong, and like the authors who submitted this gene as a record. So it has the authors' names and it has given you the taxonomy. It says like Eukaryota, Metazoa, Chordata, Craniata, Vertebrata. And it has also given you the PubMed ID. If I click on this, I think I'll get the paper. See, in the abstract it says, we have identified three new frameshift mutations in the CFTR gene in patients with cystic fibrosis. Do you guys know what is a frameshift mutation? Yeah, this is also something you will learn in genetics class, so don't stress much about this. So it just says whatever the gene that we have, this gene already has frameshift mutation in the sequence. So it is not the exact original gene, it has already mutated and it has the frameshift mutation. And you have exon details. It also has in this note section, if you can look into, the sequence consists of a new frameshift mutation in which adenine is deleted at 3 between the cytosine at 25 and thymine at 26. A cytosine has been inserted in the new mutant sequence. So it has also given you at what position there is mutation and what is replaced by what nucleo. And it was also given you the protein IDs. What if the same gene was translated? Then what would be the protein that you get? And it has given you the sequence. So, and except from the sequence, all these things are called as metadata. And three nucleotides or three bases, they make one amino acid, and you also have the translated sequence here. I think in biochemistry, if I'm not wrong, you will learn about this, right? Like single alphabet representation of amino acids, if I'm not wrong. Correct. So you have all these details, and it is also given you DNA is linear. This is also something you will learn in genetic engineering, if I'm not wrong. DNA is not always linear. You will also come across circular DNAs. And you have accession, you have version, you have different things that will be used if you want to clearly evaluate or run analysis on the data. So this is all about National Library of Medicine's NCBI. These are some of the basics that everyone usually goes through or works on. You also have some advanced features like these GEO data sets. This is nothing but gene expression omics data. For example, if you guys have heard of this, when a person is deceased, there will be difference in the proteins expressed in a healthy individual and also the proteins expressed in the deceased individual. So the translation of the genes will be regulated. It will be either upregulated where you have a lot of proteins produced, or it will be downregulated where you have only few proteins produced. And that will be used to analyze what is the biomarker for the disease. Am I making sense? Have you guys heard of biomarker? It is like what gene is causing you the problem. See, it also has this GEO data sets. It's really good. So like you can search. I think cancer is the hot topic nowadays. Yeah, you can download the data and the data is in TSV format, and it has also given you from which species the gene expression data has been collected. You have mus musculus. Can anyone guess which organism this might be? Anyone, guys? Mouse, correct. And if you click on this, you get this, and it's in the HTML format. You can change the format, and it says it has 7 samples. It has also given you some details. CD8 T cells. This is something you will learn in immunology. After learning immunology, you'll be able to better understand. It says ATF4 overexpression is seen in the sample, and it has also given you some description or the summary of what is actually happening in all these samples, and it also says the type of experiment. If you guys have heard of high throughput sequencing, it says RNA sequence data from CD8 T cells was isolated from spleens of TG or TG mice, contributor citation, etc., etc. So it has all these things, and you can download this. And I did try uh, using machine learning to find or classify the genes which were upregulated and downregulated, and try to find biomarkers for it. For that, this GEO data helps a lot, and this location from where the gene was sampled or collected will help you in understanding the phylogeny or the evolution of it. Because, for example, let's take coronavirus. So the whole world was suffering from corona, and you had different mutants or different variants at different locations in the world. What if you want to put all those things together and you want to find from where did the mutation begin and how did it mutate? Like, for example, if there was some person who came from France to India, and that mutant or that version of the coronavirus, after coming to India, it went through few mutations and it became a whole different mutant, and it spread to all the Indian community. So it helps in those kind of studies a lot. So this is about gene expression data. Are you guys interested in learning these things in depth, like applying different analysis, etc.? Okay, okay, that's great. Because I just thought during the initial few classes of machine learning, we were learning a lot. I was covering almost like 8, 9, 10 topics per day. I thought that was too much for students of first years to handle. So I thought we will just go with introduction now in the bioinformatics bootcamp, and then we'll apply analysis. And so you have all these things. We will look into these things in detail. So this was all about NCBI. You can use PubMed, those who are interested in reading research papers. or accessing research papers, PubMed is one of the best sites. I think that is all for today's session. Do you guys have any doubt? If you guys are interested, based on like how many of you are interested, I'll plan the analysis part accordingly. Again, remember, it's a kind of database where you can also submit the data. If you come to the home page, it says deposit data or manuscripts into NCBI database, where you submit the data. For example, you can select the type of sequence and from where you want to submit, etc., etc. You can choose it. I think a whole different process happens here, the approval process. And you can also download NCBI data to computer. You can use it for research, use it for analysis, you can use it. The NCBI API, yeah, this is important. I think I mentioned about the API in the drug bank as well. Do you guys remember? I told API is like a key. It's locked to, like, it's a key which can unlock this NCBI database. If I'm writing a code in Python, if I use that API and I write a query that I want this gene of this disease which is present at this location in the chromosome, it will fetch that if it is present in the NCBI. So I need not download the whole NCBI dataset. I can just use that API and I can fetch the data. It also has a GitHub repository. So I think this API will only help you if you're like, more into the coding part of it. Otherwise, it will be not of much help. So this is the basic introduction of NCBI. And you will learn a lot when we learn for when we learn different analysis. These things are cool. I think that is all for today's session. Do you guys have any doubts? And again, I'll upload all the sessions, the bioinformatics sessions in the YouTube. So if because of some commitments you guys miss sessions, you can go to YouTube and you can, watch it in 2x. If you find the session, too slow, you want to save time, you can watch it in 2x or 1.5x, however you want. I think that will be helpful as well. Any doubts? Okay. And also, please make sure to subscribe BRSI's YouTube channel. That will be like a support and so that like we can do better next time. Yes, thank you so much for attending. You guys can leave the session. Good night.